Hello and welcome to my talk. My name is Lukas Krusenbaum and I'm going to give you a quick overview of my work on rice gene bank phenomics using publicly available pan-genomic resources. First, want to address the question, what are gene bank phenomics and what is our goal? While ample genetic variation is stored in gene banks for all major crops, selecting donors from these resources for breeding programs remains challenging due to the workload of phenotyping such a large number of accessions for important traits. For rice, a large genetic resource is available in the 3K Rice Genomes project. This consists of 3,024 diverse accessions spanning all subpopulations of rice, along with genotypic data for every single one of these accessions. By phenotyping relatively small subsets of these, we want to establish genomic prediction models that are able to predict phenotypes of accessions that have not been part of the training data. This enables selection of potential donors for rice breeding programs with reduced phenotyping effort. The most common form of genetic markers used in such models are single nucleotide polymorphisms, or SNPs. These are single nucleotide variations identified between accessions. These can be generated by using the whole gap-free sequence of a single individual as a reference. In rice, the first reference sequence that was available is the Nippon-Bar sequence. While this enables SNP genotyping of a large number of accessions at reasonable cost, missing values for SNPs in individual accessions can arise due to errors in the genotyping pipeline. A commonly used approach to deal with these is to remove SNPs that have too many missing values. For example, a call rate filter of 0.8 would remove any SNP that has more than 20% of missing values. Remaining missing values are then imputed by maximum likelihood estimates. Variation of SNPs can now be encoded as continuous variables by counting the number of times the alternative nucleotide is observed in a diploid individual. Additional to SNPs, complex genotypic variation exists. In rice, stretches of sequence several thousand base pairs large spanning whole genes have been found to be absent in some accessions. Because of this, a single reference sequence cannot adequately reflect the whole sequence diversity of the rice species. Recently, 16 new reference sequences spanning all subpopulations of Asian rice have been made publicly available in the 3K Rice Genomes project. These have been used to identify new SNPs that could not be identified when using Nippon-Bar as a reference. Sequence diversity of these 16 accessions was captured by consecutively aligning these sequences to the Nippon-Bar sequence. This generates a graph structure where every vertex represents a DNA sequence. By looking at vertices not found in Nippon-Bar, we can identify large sequence variations absent from the Nippon-Bar sequence. On the left of this slide, you see a graphical representation of such a case. The light blue bars represent stretches of sequence present in Nippon-Bar as well as all other reference sequences. The orange bar represents a stretch of sequence that is approximately 10,000 base pairs long and absent from the Nippon-Bar sequence. This was first identified in the N22 sequence, which belongs to the Aus subpopulation of rice. When looking at call rates of SNPs identified in such vertices, we see that these cluster with subpopulation. For example, SNPs first identified in the reference sequences belonging to Aus on average have higher call rates in the Aus subpopulation. We can conclude that applying strict call rate filters to such SNP sets removes important genotypic information and that absence of SNPs may not necessarily be due to technical error. Accepting absence of SNPs as true requires alternative ways of encoding SNP variation, as treating SNPs as continuous variables by counts of the alternative nucleotide is no longer possible. We explored encoding only presence and absence variation of SNPs, as well as retaining variations of the nucleotide by slightly modifying one-hot encoding. For genomic prediction, we are using a multiple linear regression where an additive genetic effect parameter is estimated for every marker. As we have many more predictors than samples, this is solved by rich regression. Genomic prediction accuracy is estimated by 10 replications of 5-fold cross-validation as the Pearson correlation of measured and predicted values in the test set over all folds. We are here comparing a baseline approach using only SNPs from the Nippenbar reference of high call rate to pan-genomic approaches adding SNPs identified in sequences absent from Nippenbar. These were either SNPs of call rate larger 0.8 for the pan-G core or SNPs of call rate lower 0.8 for pan-G PEF and pan-G OHE. These were encoded as presence and absence or by one-hot encoding, respectively. Training on 9 publicly available phenotypes, we see that adding pan-genomic SNPs of low call rate substantially improves genomic prediction accuracy compared to the baseline approach for the 3 largest subpopulations Aus, tropical Japonica, and Indica. Adding SNPs of high call rate only gives small and inconsistent improvements. The majority of useful information is retained in the presence and absence variation of these SNPs. We can conclude that not applying a standard approach of call rate filtering for SNPs identified in the RISE pan-genome enables substantially more accurate prediction of phenotypes. As this is done by just modifying the pipelines of handling existing data, these improvements are achieved at no additional cost. With that, I want to thank you for your attention and all the people involved for their input and support. I especially want to thank Dr. Yongsu from KAUST for the initial hint towards these novel SNP matrices, as well as my supervisor, Professor Visuva, and the PhenoRob Cluster of Excellence for giving me the opportunity to work on this.